Acorn.
Well, thank you so much for coming today. I'm Dr. Drew Taylor, and I'm the CEO and co-founder of Acorn, and what we do is save your cells from aging. I don't think that I need to share too many notes on this. This is definitely a futuristic crowd thinking about what's coming in the future, but we are at the beginning of an anti-aging revolution. And this revolution is actually powered by our own cells. For some people, it's actually already here. We've seen celebrities receive PRP treatments, stem cell injections in the world of aesthetics, and we've also seen some of our sports heroes and athletes use stem cells, PRP, and some of these new regenerative medicine strategies to return to play faster and recover from injuries better. It's not exactly evenly distributed though, and that's an unfortunate incident because generations are going to miss out on some of these next generation therapies. As we age, our cells are aging along with us. And unfortunately, all of these next generation therapeutics that are leveraging regenerative medicine are actually using cells. And so therefore, as our cells become less effective, our potential benefit from these things is also decreasing. I saw this firsthand when I was working at Mount Sinai Hospital and working with U of T in their orthopedics division. We were essentially trying to go back to patients that are coming in for arthroplastic knee surgeries, so taking fake knees and fake hips, and take a biopsy of cells from those patients to see if in practice we could grow out the functional tissues that would be needed to replace the metals and plastics that we put in during these therapies and these surgeries. We'd actually grow them back, their own sheets of cartilage, so that they could hopefully have a lifetime solution that would last well into the future without the need of revision surgeries, could heal, respond to shear forces and stresses like natural tissues. It's a very ambitious project. My role going into that group was actually to translate some very successful animal studies into human models. And so I was going into the OR and taking biopsy of these patients and bringing them back into the lab to see if we could create these tissues for them on demand. It's a very ambitious project, and unfortunately, it did come with some challenges. And it makes sense, because when you think of it, we're going to a patient at their time of need, usually later in life, usually after some kind of disease onset, like osteoarthritis, and then asking those cells to, in the laboratory, perform at their best, and essentially recreate them those brand new tissues. Well, it was extremely age-dependent. If we had access to younger cells from younger patients, we could accomplish this. But unfortunately, in elderly patients, which we are seeing the majority of our patient base being, our hands were really tied. The easy answer would be to freeze cells from a patient before they reached that disease state and advanced age. To actually take some of those samples before the time of need. By cryogenically preserving them, we'd actually stop the effects of aging. Bring them down to temperatures in liquid nitrogen where aging stops, cell metabolism stops. This would be a dream offering for groups in the very competitive med spa, wellness, dermatology, plastic surgery markets. We see it also in sports. But unfortunately, would it be possible to actually collect the right cells without going through invasive surgery or even needles? Things like bone marrow harvesting, and some of these strategies that require an invasive surgery in themselves. On top of that, can we actually get more useful cells than we're currently collecting in most protocols, where we're doing spitting and swabs for genetic analysis? And since those cells are going to die immediately after they leave the human body, how do we actually make sure that they survive that? Transported back to an environment where they can actually be processed effectively and cryogenically preserved. Well, I'm excited to announce that that's exactly what we've been working on for the past number of years. Developing an efficient way for us to intercept aging and disease. Actually take a patient's sample non-invasively, involving just targeting hair follicles from the back of their scalp. Painless, non-invasive, and very easy to do. Furthermore, take those cells and have them survive that journey from the clinic through to our cryogenic suite, which is at one of Toronto's largest hospitals downtown, and be cryogenically preserved and safe there for that patient's lifetime. This is the first non-invasive and accessible way to preserve skin and hair follicle stem cells for future treatments. A little bit about why we target the hair follicle. Well, this is a very rich variety of cells. It almost feels like the hair follicle was designed specifically for this. You've got keratinocytes and fibroblasts, two different germ layers in our bodies. We have high percentages of adult stem cells. They're highly expandable and reprogrammable, so they can be used within areas for skin and hair regeneration, but also in areas outside of that, like sports medicine and beyond, which we'll get into. But they can also be collected non-invasively and painlessly, but most importantly, these cells are uniquely your own. You don't have to think about future complications because of immunosuppressants or rejections from donor cells. In fact, Acorn in our lab, we've been able to take a single hair follicle from a patient and turn that into a pluripotent stem cell, further pushing it into pancreas, progenitor cells, and other cell types so that a patient can have really any cell type that they require on demand in the future. This is a very exciting strategy that we have really been pressure testing over the last number of years, shipping samples across the country in clinical trials, ranging from Vancouver to Montreal, and shipping those cells over thousands of kilometers to our central facility in Toronto. We've been able to accomplish keeping those cells alive during days of transport instead of worrying about minutes or hopefully even hours. We've built it a world-class processing analysis and cryogenics facility, and again, I said it's embedded into one of Canada's top hospitals in downtown Toronto, and this is where those samples are then divided, put into multiple vials and aliquoted so that a patient has multiple vials of their own younger cells for them on demand for the rest of their life. These cells then act as a reservoir, as a resource for that patient in a number of different ways. Initially, some of the most near-term applications are in the area of aesthetic medicine. So thinking about rejuvenating skin and regrowing hair. These cells can act as a factory where we can then produce growth factors and the building blocks of our connective tissues and skin on demand for patients to use in regenerative therapies throughout their life. In the medium term, these cells can actually be reintroduced to our bodies, providing a younger version of ourselves to influence that older microenvironment around those cells and actually act as performing cells in our tissues that drive that rejuvenation of that tissue. Long term, we see these cells plugging in and merging with technologies like iPSC and CRISPR, where we're able to actually dial those cells to create whatever cell type we need for a given therapy, even edit out disease in the genetic code through CRISPR. And we have a wide range of projects that are going on with some of Canada's top institutions, including creating neuronal cells and blood-brain barrier cells with the NRC from a human hair follicle sample, working with the University of Toronto to create a simple solution to create iPSC cells on demand for patients autologously, working with Mount Sinai Hospital to then turn those samples into pancreas progenitor cells, IMSCs, kidney cells, and then working with groups like University of Ottawa and McGill University to perfect the analysis of that whole end-to-end solution for a patient. We've assembled a pretty great team from a very diverse background, and it's very exciting for us to come and share some of our recent work today with this group. Being in the healthcare sphere, it's always an exciting opportunity to share what we're doing with people in other areas like esports and gaming and blockchain, and I think there's a lot of cross opportunities. But what I'm really excited about is the people that we see working Acorn, I see reflected in everybody at this conference, because ultimately this is a conference for futurists. This is early adopters of next generation strategies and technologies that are going to shape the future for us. So thank you so much for allowing Acorn to be a part of it, and would love to connect with people during or after the conference further. Thank you again.